Hello everyone, myself Dr. Manjula S, working as Assistant Professor and Head of the Department of Botany in Government First Grade College, Ramnagara. In this video, I am going to explain secondary growth in dicot stem. This topic is prescribed by the Bangalore University in first BSc, second semester Botany syllabus. I am going to explain this topic with the following content, which are as follows. Introduction to secondary growth in plants, followed by role of meristems in secondary growth, an overview of secondary growth in dicot stem, definition of anomalous secondary growth, and anomalous secondary growth in Dracena, which is an example of monocot stem, followed by anomalous secondary growth in Bohirarhea, which is an example of dicot stem, and I am going to conclude this topic with significance of secondary growth in plants. Let me start with the introduction. As we all know, plants exhibit two types of growth. One is primary growth and another one is called the secondary growth. Primary growth is the growth in length of a plant which results in development of lateral appendages, whereas the secondary growth is the formation of secondary tissues from the lateral meristem, which increase in the girth of a plant or increase in the diameter of the stem. In woody plants, the secondary tissues constitute the major part of the plant, which take part in providing protection, support, and conduction of water and mineral nutrients. Secondary tissues are formed by two types of lateral meristems, which occur in the side walls of the plant, like vascular cambium and cork cambium. The vascular cambium produces secondary vascular tissues like secondary xylem and secondary phloem, whereas the cork cambium produces phenlogen and peridot. The secondary growth usually occurs in perennial gymnosperms and also in dicots such as trees as well as shrubs. So in this slide, we can see the primary and secondary growth in a plant, where the primary growth is mainly due to the presence of apical meristem, which results in increase in the length of a plant. This is the primary transverse section of a stem, shows the primary growth, where we can see the epidermis followed by cortex and the stellar region, where there are vascular bundles with cambium and large pate. During the secondary growth, it is mainly taking place due to the presence of lateral meristem, which are present in the side walls of the plant. The lateral meristems are of two types, vascular cambium and cork cambium. The vascular cambium is going to produce the secondary xylem towards inside and secondary phloem towards outside, whereas the cork cambium present in the cortex, it is going to produce the phellum towards outside and peridot towards the inside. As I already told, there is an involvement of meristems in bringing about the growth in a plant. So, the apical meristem is going to form three different types of initials like protoderm, ground meristem and procambium. This protoderm, during the primary growth, it is going to produce a primary tissue called epidermis, which will give protection to the internal organ, whereas the ground meristem, during the primary growth, produces a primary tissues called pit and cortex, whereas the procambium, during the primary growth of a plant, it produces primary phloem as well as the primary xylem. Whereas in case of secondary growth, it is mainly due to the activity of the lateral meristem. The lateral meristems are going to be form two initials called cork cambium and vascular cambium. This cork cambium later divide and form cork and phelloderm, whereas the vascular cambium divide and form the secondary phloem and secondary xylem, where the secondary xylem is commonly called as wood, and this cork, phelloderm and secondary phloem together known as the bark of the plant. So let me move on to an overview of secondary growth. The secondary growth in plant is mainly initiated by the cell division in lateral meristems, which are present at the side walls, which adds width to the older areas of the plant, which results in increase in the girth of a plant. It arises in a woody plant where the primary growth has completely ceased, which usually occurs during first and second year of the plant's growth. The process when differentiated cambial cells revert back to undifferentiated cells and result in the formation of two lateral meristems like vascular cambium and cork cambium, which later divide repeatedly to produce new growth called the secondary growth. Vascular cambium formed from procambial cells produce secondary vascular tissues like secondary xylem and secondary phloem in the stellar region, whereas the cork cambium, which is technically called as the phloem, formed from the parenchyma cells of the cortex, is going to produce new dermal tissues like phloem and phellum in the cortex region. So in this diagram we can see this is the primary growth in the stellar region, whereas this exhibits the secondary growth. After the secondary growth, there is a complete ring formation in the stellar region. So in this diagram, we can see the initial formation of the secondary growth where the cambium present between the xylem and phloem is called intravascular cambium, whereas the cambium present in between the two vascular bundles is called intravascular cambium. Due to the division and activity of this both intravascular cambium and intervascular cambium, after the secondary growth, there is a formation of complete ring, which is otherwise known as the growth ring. In the secondary growth, after several years, we can observe there is a large amount of secondary xylem is produced inside of a plant, whereas the secondary phloem is produced towards the outside or cortex region. And in the cortical region, due to the activity of cork cambium, the phellum is going to be produced towards the outside, whereas the peridome going to be produced towards the inside of the cortex. Secondary growth in vascular region is going to produce two different types of tissues. First one is called the secondary xylem. Secondary xylem is commonly called as wood. It forms the bulk of the stem or major part of the stem, which largely consists of dead cells. The more recently formed layers of the secondary xylem involved in conduction of water and mineral nutrients, whereas the second type of tissue is called the secondary phloem, which is commonly called as bast. It forms a narrow circle around the outer side of the vascular cambium. It does not grow in thickness because the primary and the older secondary phloem present on the outer side get crushed with the secondary phloem present on the outer side, and the development of new functional phloem, therefore rings are otherwise known as annual rings are not produced in the secondary phloem. More recently formed layers of the leaving secondary phloem involved in the conduction of food. In order to provide increase in girth and to prevent arm and the rupturing of the outer ground tissues due to the formation of secondary vascular tissues, dicot stem also produces a special type of cambium called cork cambium, which is technically known as the phloem in the outer cortical cells. Rarely, it may also arise from the epidermis or hypodermis, which is otherwise known as outer cortex, or phloem parenchyma also. The secondary tissue produced on the inner side of the phloem is parenchymatous in nature or collenchymatous. This is called the secondary cortex, which is technically called as the phloem, whereas the phloem is going to produce cork or phellum on the outer side of the plant, which prevents the loss of water by evaporation. Cork is light in color, compressible, non-reactive and sufficiently resistant to fire. As I already told, there are basically two types of dermal tissues in vascular plants. First one is called epidermis and the second one is called the periderm. The epidermis is formed during the primary growth of a plant, whereas the periderm is formed during the secondary growth by the activity of cork cambium present in the cortical region, which consists of three important tissues like the phellum, which is otherwise known as the cork, phloem, otherwise known as the secondary cortex, and phloem is also known as cork cambium. The phellum, which is going to form towards the outside, it consists of dead cells when mature, whereas the phylloderm, which produced towards inside, consists of thin layer of living parenchyma. So in this diagram, we can see the portion of transverse section of dicot stem showing the secondary growth, where there is a pith and this is a vascular ring. After the secondary growth, there is a production of secondary xylem towards inside due to the activity of cambium, and secondary phloem towards outside. And in between the two vascular bundles, the medullary rays are replaced by xylem ray and phloem ray, whereas in the cortical region, due to the activity of carp cambium, it divides and redivides and it forms the cells towards inside, that is called the phylloderm, whereas it is going to produce the cells outside, that is called as the phellem. And due to the repeated formation of the cells or addition of number of cells, a pressure is created which results in the opening of the outer epidermal layer, that is called the lenticell, which will meant for exchange of gases. So as I already told, lenticels are aerating pores in the bark region of a plant. They appear on the surface of the bark as right scars containing oval, rounded and oblong depressions. They occur in woody trees, normally formed in areas with underlying rays for facilitating gas exchange. A lenticell is commonly produced below the stomata of the epidermis. Its margin is raised and it is formed by the surrounding cork cells. It is filled up by loosely arranged, thin-walled, rounded, suberized or unsuberized cells called the complementary cells. They enclose intercellular spaces meant for gaseous exchange. So in this diagram, we can see the stages of secondary growth in a stem, where there is the activity of the cambium. This cambium is called intravascular cambium, which is present within a vascular bundle. It is going to cut down the secondary xylem inside and secondary phloem outside. And this cambium is called intervascular cambium, which is present between the two vascular bundles. So it will cut down xylem rays and phloem rays. And after the secondary growth, the medullary rays are filled by xylem ray and phloem ray. And when the complete ring is going to be formed during the secondary growth, which is otherwise known as the growth ring. At the time of initiation of the secondary growth, the cambium is going to divide cells towards inside. The cell which is formed towards inside is called xylem mother cell, which later divide to form the secondary xylem, whereas the cell which is formed towards the outer or outside is called the phloem mother cell, which divide to form the secondary phloem. So, as a result of secondary growth, when the complete ring is formed, one more cambium is become active and it is going to cut down the secondary xylem inside and secondary phloem outside in the ciliar region, whereas in the cortical region, due to the activity of the carp cambium, it is going to cut down the two group of cells. The cells which are formed towards inside are called as the phylloderm, whereas the cells formed towards the outside are called as phellem. So in this diagram we can see the number of annual rings formed as a result of the multiple secondary growths in a stem. So this is a diagram, a dicot stem showing secondary growth where it well underwent the secondary growth, where we can find the pith, and this is a large and massive secondary xylem produced as a result of secondary growth, and this is secondary phloem, and these are called the ray initials formed as a result of the secondary growth. And in the cortex region, due to the activity of carp cambium, the secondary tissues are formed. So I am going to conclude the secondary growth in plants, which is mainly due to the activity of cambium. The cambium are lateral meristems are of two types, vascular cambium and carp cambium. During the secondary growth, this vascular cambium is going to form two initials called fusiform initials, which are vertically oriented cells, whereas the ray initials contain horizontally oriented cells. These fusiform initials divide and form the secondary xylem towards inside and secondary phloem towards outside, whereas ray initials, which is present in between the two vascular bundles, that divide and form two different types of tissues called xylem phase and phloem phase, which is taking place at the stellar region. Whereas in the case of cortical region, the cardiac cambium is going to be developed. This is otherwise known as the phellogen. This cardiac cambium is going to form two important tissues like phellum or phellodome, which are together known as the periderm, which is replaced the epidermis after secondary growth. So now I come for the definition of anomalous secondary growth in plant. In many plants, the pattern of secondary growth thickening shows deviations from the normal type. Hence, the term anomalous is used here, which is different from the normal growth. So, it is more common in tropical plants where it is not an anomaly or disease. Instead, it is an adaptation of a plant to that particular environmental habit. Here, normal cambium, which is situated in the abnormal position, it behaves irregularly, resulting in the abnormal arrangement of vascular tissues. The normal cambium does not develop, or if it is present, it is going to be replaced by additional or accessory cambial rings. Secondary growth is usually absent in case of monochord because the monochords have closed vascular bundle. Closed vascular bundle means there is an absence of cambium. However, a very few plants like yucca, aloe, agave and dracaena exhibit anomalous secondary growth. The secondary growth thickening in dracaena is a classical example of anomalous secondary growth in monocot plant. So let me explain the primary structure of dracaena stem. It has the following structures like epidermis, hypodermis, ground tissue and vascular bundles. The epidermis is the outermost single layer made up of parenchymatous cells which will give protection to the internal organ of a plant, whereas epidermis is followed by hypodermis, which is otherwise known as outer cortex. It is present below the epidermis and made up of sclerenchymatous cells. Here we can observe the hypodermis made up of the sclerenchyma. And the hypodermis is followed by the ground tissue, which is undifferentiated and it is parenchymatous in nature. Inside the ground tissue, there are a number of vascular bundles scatteredly arranged in the ground tissue which are closed type. That means there is absence of cambium. This is the dracaena plant. This is the transfer section of dracaena stem in its primary growth. In dracaena, secondary thickening is initiated by the formation of special meristematic zone. Meristematic zone means the cells which are having actively dividing capacity. As a result of their division, a multi-layered cambial ring is differentiated from the inner cortical cells lying outside the vascular bundle. The shape of these cambial cells varies and the activity of the cambium is abnormal in nature. So the cambial ring cuts the large number of cells to the inner side and very few cells to the outer side. The cells which produced external to the cambium are parenchymatous in nature, whereas the cells produced inner to the cambium are partially parenchymatous and partially vascular in nature. The parenchymatous cells produced internal to the cambium develops into a lignified conjunctive tissue, whereas the vascular tissue produced internal to the cambium, the vascular tissue produced inside to the cambium is differentiated into secondary vascular bundles. So this is the section of dracaena stem where we found the activity of cambium is initiated in the cortical zone. As a result of the cambial activity, it is going to divide and cut down large number of cells towards inside and very few number of cells towards outside. The cells which are cut down towards inside are of two types. One is parenchymatous tissue that will later called as conjunctive tissue, and the second one is called the secondary vascular bundle, which are formed outer to the primary vascular bundle. The cells of the inner cambial ring are participated in the formation of secondary vascular bundles which are concentrate and amphivasal. Concentric means one after the other. Ring is going to be formed, one ring followed by another ring. That condition is called the concentric ring, and the vascular bundles are amphivasal. Amphivasal means there is a central phloem surrounded by the xylem. Usually, the secondary vascular bundle arises from a single initial cell called vascular bundle initial, which divide by two anticlinal division to form 2 or 3 cells. All these cells then undergo a periclinal division to form a peripheral cells. Later on, divisions are irregular to form a mass of cells. In this mass of cells, the centrally placed cells get metamorphosis into the phloem tissue, whereas the peripheral cells get metamorphosis into the xylem elements. So, in this slide we can see the secondary growth in Dracaena stem. This is the ground plan of TS of Dracaena stem, where we can find the outermost layer is called epidermis, followed by the cortex, and it is the stela region or ground tissue where the vascular bundles are scattered. Since it is the secondary growth of these are the primary vascular bundles. After that, there is activity of the cambium initiated in the cortical zone, which results in the formation of number of secondary vascular bundles and also conjunctive tissue. In the cortex region, the activity of cambium results in the formation of the cork cells, which are towards outside. So, this is a part of this stem, a portion enlarged in the Dracaena stem, where we can find these are the primary vascular bundles, and due to the activity of cambium, which is initiated, multi-layered meristematic tissue is formed in the cortex region, which is going to divide and cut down large number of cells towards inside and very few cells towards outside. So, the large number of cells towards inside include the conjunctive tissue and the secondary vascular bundle, whereas the few cells cut down towards outside are parenchymatous in nature, and the activity of cambium in the cortex region is also going to form the cells called cork cells. Thus, the newly formed secondary vascular bundle consists centrally placed phloem surrounded by the xylem. That condition is called amphivasal vascular bundles. The cambium then cuts parenchyma cells internally, which pushes the vascular bundle towards the central region. After some time, the cambial ring again behaves abnormally to produce another ring of vascular bundles. This process continues, and thus many concentric rings are arranged one after the other. The last ring of vascular bundle is embedded in the mass of lignified conjunctive tissue. A cork cambium may also develop at the cortex region, which can later produce the bark of the plant. So, let me explain the secondary growth in Bohirabia, which is an example of dicot stem. Bohirabia diffusa is a medicinal plant belonging to the family Nyctaginaceae. So, in its primary structure, it has the following. First, outermost layer is called epidermis. It is the outermost single layer made up of its parenchyma and has numerous multicellular heads called trichomes. Epidermis is followed by second layer called hypodermis. It has 2 to 3 layers of thickness and composed of thick wall collenchyma cells. Hypodermis is followed by inner cortex composed of parenchyma cells with plenty of intercellular space. And the fourth layer is called endodermis, which is present but is not distinct or conspicuous, whereas the pericycle is 2 layered, and next to the pericycle there is a vascular bundle. It contains 3 rings of vascular bundle. The 2 large centrally placed medullary vascular bundle we can see in this diagram. There are 2 large vascular bundles which are facing each other, and followed by the second ring, a middle ring of 6 to 14 loosely arranged medium-sized vascular bundles, and the outer ring consists of 15 to 20 small vascular bundles, where all the vascular bundles are conjoined, open and endark in nature. Conjoined means the xylem and phloem are together in a single vascular bundle, whereas open means the cambium is present between the xylem and phloem, and end arc means the protoxylem facing towards the pit. Coming to the anomalous secondary growth in case of Bohirevia, here the secondary thickening occurs by the formation of successive rings of accessory cambia. After the primary growth, the vascular bundles of middle ring increase little bit in size to a limited extent by the activity of their intrafascicular cambium. There is no secondary growth is observed in the second layer or middle layer of the vascular bundles, whereas the outer ring exhibits the secondary growth. The fascicular and interfascicular cambium of the outer ring joins to form a continuous cambial ring. This cambial ring produces internally secondary xylem in intrafascicular region and lignified conjunctive tissue in interfascicular region. Externally, the secondary phloem in the intrafascicular region and parenchyma from the interfascicular region. The activity of this cambium is very short. After some time, its activity is going to be ceased. So, in this diagram, we can see this is a transverse section of Bohirevia stem with secondary growth, where we can see the large inner two vascular bundles which shows secondary growth. Only it will increase in the size, whereas there is no secondary growth in the middle layer, and the secondary growth is prominent in the outer ring, where due to the activity of cambium it is going to form two types of tissue, secondary xylem and secondary phloem, and this is the conjunctive tissue, and this is called the secondary cortex of parenchyma, which is called parenchymatous in nature. So where we can find the outermost region is called epidermis, followed by a region of hypodermis, which is made up of the collenchyma cells, and due to the activity of the cambium in the outer ring, it is going to form the secondary xylem towards inside, which are embedded inside a conjunctive tissue, and secondary phloem towards outside. And this is the ground tissue where the vascular bundles are embedded. A new cambium arises by joining the secondary parenchyma cells lying opposite to the phloem, which also behaves similar to the previous cambium. It produces secondary xylem alternating with the lignified conjunctive tissue towards inside. During the secondary growth in Bohirevia, the activity of cambium results in production of secondary xylem and conjunctive tissue towards the pit, whereas the secondary phloem and parenchyma towards the epidermis. After some time, the activity of the second ring of the cambia also ceases, and additional cambia are differentiated external to the previous one. The phellogen, which is nothing but the cork cambium, is initiated in the cortex region or outer cortex, which is otherwise known as hypodermal region, which produces the phellum towards outer side and phellodum towards the inner side. So, in this diagram, this is a portion of transverse section of Bohirevia stem, where we can find the secondary growth. So, these are centrally placed large-sized vascular bundle which are facing opposite to each other. So, it exhibits the xylem towards inside, phloem towards outside, and these are the medullary bundle, and there is a cambium present in between the vascular bundle. There is no secondary growth is exhibited in the second ring of vascular bundle. Only the secondary growth is exhibited in the outer vascular bundle, where we can find the activity of cambium, which bring about the division and redivision, and it forms the secondary xylem towards inside, which are embedded in the conjunctive tissue. The conjunctive tissue is nothing but the parenchyma cells produced by the cambial ring, which are towards inside, whereas this cambium produces secondary phloem towards outside and parenchyma, which is going to form the secondary cortex. In this hypodermal region, the cork cambium become active. It is going to form the phellum or bark towards outside and phellodum towards inside. The phallodome is otherwise known as the secondary cortex. So again, this is another diagram explaining the growth of secondary growth. In case of Bohirabia stem, this is a large medullary vascular bundle followed by a second ring, and in the third ring, this is the secondary xylem formed after secondary growth, and this is the secondary phloem formed after secondary growth. This is the conjunctive tissue. Hence, the number of vascular bundles are embedded inside the conjunctive tissue after the secondary growth takes place in this Bohirabia stem. So, the process of accessory cambium formation repeated like this, 4 or more concentric vascular bundles are formed which embedded inside the conjunctive tissue. They separated by a thin wall parenchymal zone to give the appearance of growth rings. In this diagram, we can observe the number of concentric rings are formed as a result of secondary growth in case of Bohirabia stem. So now, I am going to conclude this session by telling the significance of secondary growth. The secondary growth adds to the girth of a plant or increase in the diameter of the plant. It provides support to increasing weight of the aerial growth. Second, secondary growth produces a corky bark around the tree trunk that protects the internal part from abrasion, heat, cold, and microbial infections. Third significance of secondary growth is it adds new active conducting tissues for replacing the old ones which will become non-functional after some time, as well as for meeting increased demand for long distance transport of water and organic nutrients. The plant showing secondary growth can grow and live longer compared to other plants. The last important of secondary growth is it is going to produce the massive wood which represents the secondary xylem, is a very important product of secondary growth which is of commercial value. So with this significance, I am going to complete this topic. Thank you.